Alright guys, good morning. What we're going to do today is I'm going to run you through at least the notes on meiosis. These are already posted on Google Classroom, but from talking to a couple of you here and there this morning with some meetings, it seems like we need some clarification. Alright, so the main goal of meiosis, which is different than mitosis, is we're trying to produce haploid cells, which means half the amount of chromosomes. So, normal human, 46 chromosomes, but now you have sex cells, you only want to have 23 chromosomes, which makes sense. During fertilization, a sperm and an egg have to meet, and when they fuse and we form that new zygote, it better have 46 chromosomes, which means we needed to cut things down to 23 and 23. So our main goal in meiosis is we're going to make a haploid cell. And what that means for us is we have to go through 2 rounds of cell division. So mitosis, we just copied our DNA and divided once. But in meiosis, we got to copy the DNA and divide once and divide again. And so there's some fundamental differences here, and there's also some fundamental things that are going to occur during meiosis that don't happen during mitosis. The cell types that are going to be using this are going to be sex cells. So gentlemen, sperm, ladies, egg. And that's pretty much all we're looking at. The steps are going to be the same as mitosis. I'm still going to have prophase, metaphase, anaphase, telophase. I'm still going to have to get rid of my nuclear envelope and have spindle fibers and centrioles and move things around. So none of that's really going to be different. Only now I'm going to have 2 rounds. So I'm going to have prophase 1 and prophase 2. I'm going to have metaphase 1 and metaphase 2. And you'll see as we get to it here how that's going to work. Now, one of the major fundamental differences between mitosis and meiosis is this idea of synapses and crossing over. So you guys are all familiar, because we've all done karyotypes, that each of you has genetic information from your mom and your dad. So from your mom you have a chromosome 1, and from your dad you have a chromosome 1, and they're homologous chromosomes. Well, in meiosis, one of the unique things that's going to happen is these 2 chromosome 1s, or chromosome 2s, or they're going to find each other. And when they find each other, they're going to line up next to each other in a nice, unique little pattern, right? So here you can see that's dad's chromosome and there's mom's chromosome. And it's called a tetrad because you have 4, 1, 2, 3, 4 chromatids lined up next to each other. And remember, tetra, Greek prefix meaning 4. So we're going to have this tetrad, and what's going to happen is some of dad's chromosome, the arm is going to kind of cross over mom's, and when they cross over, they can kind of break off and get some new genetic information. So as you can see, we get this crossing over, and now we have 1 chromosome here, 1 chromatid from dad, that will be all dad's information. It's all orange. And then you get another one here, well, it's partially dad and partially mom, and you can see there's one that's partially mom, partially dad, and then all mom. So what we're getting here is genetic variation, and each and every one of you does this every time you make sperm or egg. Your parents did it as well. If you come from a family where you have 5 or 6 siblings, I'd be willing to bet if you all have the same parents that you don't all look alike. And it's because every time that dad made sperm, he had a unique variation of this crossing over, and the same thing with mom. So we get this genetic variation, and that's important. This is why we all don't look exactly alike, even though you might come from a big family and have lots of siblings. So this is an important thing, and this all happens during basically prophase. Prophase 1 of meiosis is when this is going to be occurring. Okay. Now, what this means is that crossing over gives us all these new combinations of genes. It's what's known as a genetic variability or genetic variation, which is why your family members don't look quite alike. The other thing that's nice is that when you're making sperm and egg, remember you have all this information that came to you from your dad and all this information that came to you from your mom. Well, all of dads don't line up here and all of moms don't line up here. They can mix and match wherever they want. So they don't have any kind of particular place where they have to line up. And this is what's known as independent assortment. So when you're making sperm or egg, you might have, this would be a chromosome from mom and this would be a chromosome from your dad, and here's one from your dad. And they can mix and match in any way they want, which again is going to add to that genetic variation and genetic variability that you can witness within families where you have siblings that have the same parents. Okay. Now, this is just a quick overview picture of what we got going on through meiosis. And again, there are 2 rounds of it. You got meiosis 1 and meiosis 2. The key is we have to copy our DNA before we go ahead and do the cell division. So here you can see chromosomes. Let's say the red one is from mom and the blue one's from dad. Well, they had to copy. So there you can see they copied. Now, what they also did during this process is you can see the little color shift there, right? Some of the reds got some blue on it, and some of the blues got some red on it. They crossed over. Now what you also notice is it's only the arms that are right next to each other that are going to cross over. And it's because of that proximity, because they are right next to each other, they can break off and cross over. You're never going to see any kind of crossing over on any of these outside chromatids. Okay, so there's our first round of cell division, meiosis 1. So we went through prophase 1, metaphase 1, anaphase 1, and telophase 1, and we get down here to these two cells. Now I got to go through meiosis 2, but before I can do that, I got to make sure I don't copy my DNA. Remember, I'm trying to cut my chromosome number in half. So here I don't want to copy my DNA, I just want to separate. And when I separate, because of this crossing over, there I get one that's all red, there's one red with a little bit of blue, blue with a little bit of red, and all blue. I get 4 unique cells. None of them are identical, and because of the crossing over, you can see that we get some of those changes. Again, very, very important concept to understand. That's a major difference between meiosis and mitosis. Okay, so here's just kind of the big picture of what's going on. We still have to go through prophase 1, so I still have to get my centrioles, I still have to get my spindle fibers, I still need to break down the nuclear envelope, I still need to have my chromosomes condensed. All of the same stuff that we did in mitosis is going to happen in meiosis, with the exception of now we got to have that tetrad in the crossing over, all right? And we're still going to line up in metaphase, we're still going to separate everything, and we're going to go to telophase. But we have a second round. Those 2 cells that we get out of meiosis 1, we got to take both of those, and they have to go through the process again as well. So I should end up with 4 unique individual cells. That's the whole goal, okay? And each one of them should be a haploid cell. Only 23 chromosomes, and those chromosomes shouldn't be all sorts of crazy mixed up now with the genetic variation due to that crossing over in the tetrad and synapses. Now, one last little thing. Ladies, you make eggs, you only do it about once a month. Fellas, we make sperm, we do this all the time. And the reason is, ladies, when you make an egg, you've got to package everything for a new life. All the cytoplasm, all the organelles, which means human egg is going to be the largest cell in the human body. And on the flip side of that, the sperm, all we have to do is package genetic information, DNA. So it's going to be one of the smallest cells in the human body. Well, to get a large cell in the human body means we need to do an uneven division in meiosis 1. So, ladies, when you're making eggs and you have that first initial cell in your ovaries and it has to go through this process, the first division is going to be an uneven division. And all of the cytoplasm and organelles and all the nutrients are going to be packed into this one cell over here. Now, we go through our second round of cell division, and again, you can see there's an uneven cell division. We're trying to just jam everything we need to create a life into this one cell. So ladies, you start with one and you end with one viable egg. These other 3 things, called polar bodies, they're not viable. They will never, be fertilized. They will not form an egg. Predominantly, your body just reabsorbs that material and uses it for something else. Okay, fellas, we do things differently. We just package DNA. So we start with one cell and we end up with 4 unique individual cells at the end. We do this day in and day out. It's not like a once a month thing for us. The last thing to take a look at is just the differences between asexual and sexual reproduction, which you guys can read this, and most of you sophomores, juniors in high school, you pretty much know at this point the difference between these two, so I'm not going to belabor that. Alright, if you have any questions for me, you can always post something in the comments on Google Classroom, or you can just straight up shoot me an email and I'll get back to you. For those of you that did attend the meeting today online, like I said, all of your tests from here on out are going to be open note. So if you have questions, ask, but again, you're going to be able to use this stuff to help you. Alright, I hope that was helpful. If you need anything from me, get a hold of me. Alright, have a great day.